Hey everyone, this is Dr. Mikola Rashik of Maragenomics making another quick update on mRNA vaccines because recently a paper came out that has made crazy bombastic claim, just absolutely a shocker because it's the first publication, scientific publications that's peer-reviewed that makes a claim that mRNA vaccines can be converted back into the DNA. Really, really shocker because for the last more than a year and a half, all the experts and fact checkers around the world have been telling us otherwise, that that's not possible. And here we see a first example that indeed is possible. However, it has been achieved under unusual circumstances. So I do want to unpack this, tell you my interpretation of this scientific information, especially since from what I've seen right now, there's a lot of chatter on the internet going on and a lot of interpretations that I see, I personally would not agree with. So I wanted to tell you what I think. But this is really complicated topic. I need to give you a bit of a background as per usual. And first of all, let's make a claim. What did they observe? So yes, they did show supposedly that the mRNA vaccine can be converted back into DNA. They did not show that it can be reinserted back into our genomes. So it's very, very different. So if you're vaccinated, do not panic. Do not think that this is happening. There is no proof of that. If you're unvaccinated, don't start saying, see, I told you so, because what is demonstrated is because that was shown in very unusual circumstances. So first, let me give you a bit of a background. The cells that these authors used were liver cell lines, but that was a cancerous cells. And these cancerous cells, normally cancer does not follow normal rules of behavior. This is why it can be cancerous. So unusual things can happen in such a cell line that does not behave even remotely close to what our normal healthy cells are like. So that's the first important observation. And the second one is the fact that they were able to suggest that it's a very specific mechanism that might be converting the RNA back into the DNA. I talked about this mechanism. It's called line 1. I talked about it in video number 10 previously, so check it out when I discussed how possibly some authors have claimed that SARS-CoV-2, the virus, might be inserting itself back into our genomes. But let me give you a little bit more information on this. So line 1 stands for long interspersed nuclear elements 1. So the name actually gives it away. Interspersed nuclear elements, it means it's a gene that is dispersed all over our genome. And it's a selfish gene because it's this very, very unusual gene that can actually copy itself back from RNA into DNA and reinsert itself back into our genome. Throughout our human evolutionary history, it has done it so much that almost somewhere between 15 to 20% of our genome is just that gene interspersed throughout our genome. You would want to perhaps wonder as to why this event is happening. And the only possible explanation is that this has been retained for evolutionary purposes, because having this unusual jumping gene element allows genetic material potentially to recombine in unusual manners, and that can at times drive the evolution of our species. Normally, it causes problems. As I already alluded to, it can be activated inside cancer, so it can promote cancer development. It has also been involved in disease development, but I will also give you one unusual example where this element might have been used by our cells in order to progress the proper function of those cells. And I will also tie it to vaccines. Very unusual observations that have been discovered in 2018, which means all of the fact checkers that have been saying, no, it's impossible to convert back from mRNA into DNA. Well, they should have known better before they ever said that, because the information of how our potentially natural cells might be using these selfish genes, that's been already around since 2018. So first, let me give you background on this selfish gene. So how does it work? Normally, when a gene exists inside our DNA, in order for the gene to be used, it has to be copied into RNA. Then RNA will exit the nucleus and it's used as a template or a blueprint in order to produce a protein. And a protein, think of protein as a tiny molecular robot that will do all the work inside our cells. So that's how I think of robots. Robots basically make the cell function. You need the blueprint to build the robot. That blueprint is the RNA, but you don't go backwards. So you cannot go from protein to RNA. And normally you do not go back from RNA to DNA unless you were to have a very specific protein, tiny little molecular robot that could do it. It does exist in nature. It's called reverse transcriptase. So as an example, HIV patients, they have that because HIV, the virus, produces one of these. It has a template to produce reverse transcriptase, and this is how HIV can insert itself into our genomes. And this is also why HIV patients, amongst the cocktail of medications that they have to take, one of the medications they have to take is a reverse transcriptase inhibitor. So it's an inhibitor that will stop that protein that can reverse transcribe RNA back into DNA from properly functioning. Now, we humans normally do not have it except this one gene, the selfish gene, the line 1 gene. So how does it work? That gene is copied into RNA. The RNA is then exits the nucleus, and inside our cell, the environment outside the nucleus, inside the cell, it's called cytoplasm. Inside the cell, the RNA is used as a template to produce the protein robot. And here's the unusual part. This protein robot is selfish in that it doesn't go and does anything for the benefit of the cell. It goes back, grabs its own RNA, because remember, there will be plenty of RNA produced. It will grab its own RNA and sneak back into the nucleus. Now, one of the components of this protein robot, it has the ability to cleave the DNA. It will just cut one of the DNA strands and then use the RNA as a template to reverse transcribe it back into the DNA, and while inserting that new DNA right into our genome. So it's very similar to how CRISPR-Cas9 system works, if you're familiar with that system. So it's almost like we have our own version, and this is how this selfish gene can go back and read itself into our genomes over and over and over, many times over. So you can see if you dysregulate its function, how it could be promoting potentially something like cancer growth, because you could be really distorting proper genetic material. So in the context of these specific selfish genes, apparently once in a while, extremely rarely, it could supposedly grab other mRNA, not its own, and bring it back into DNA. But that was assumed basically as impossible or so extremely rare that it's negligible. But I'll tell you right away, after we go through the publication, that perhaps we should have known better. So now let's take a look at the paper that these authors published that show how mRNA vaccine is converted back into DNA. So let's jump right in. Here is the publication. And that's the link. Here's the title. And we're going to go through the data. We're going to focus on this figure right here. And what this figure shows is that these authors were able to show that when they took this cancer cell line, they were able to successfully transfect it with vaccine. That's all that this figure shows. So that shows you that once they took the cells, these cancerous liver cells, and exposed them to the vaccine, specific quantity, that's what the V1, V2, V3 means. Those are simply different amounts of vaccine, that the vaccine was taken up, and they measured the success of that by doing a PCR on the vaccinal RNA. So basically what PCR does, the way PCR works, this is the same PCR test that we've been using to track who's been infected versus not throughout the entire pandemic, is that PCR can copy a specific fragment, genetic fragment of interest over and over and over. So they did the same thing. they focused on very specific fragment of interest and they copied it over and over and over. And that's how they were able to show, hey, look, these very tall bars that you see in this figure, this shows us that the RNA from vaccine is inside the cells because we can copy it over and over and over. One of my big issues and concern in terms of a potential plot hole in this entire paper is the fact that their controls, meaning cells that were not exposed to the vaccine, as you can see, control right here, they also show presence of that RNA being copied over and over by PCR. So just clarification, normally you don't do it with RNA, you do it with DNA. So when they transfect the cells with RNA, then they isolated the RNA, they had to convert the RNA outside the cell into the DNA, and that's what they amplified. So I just want to make that clear, but nevertheless, they were able to show that for whatever reasons, they also noting that controls also have these vaccines inside them, despite the fact that we're not exposed. So to me, that's a bit of a problem because perhaps they claim that they're seeing vaccine being converted back into DNA. It might not be the vaccine. It might be whatever contamination that is present in these cells that are not even exposed to the vaccine. So what is giving this signal? It's much, much smaller than the cells that were exposed to the vaccines, but nevertheless, you can clearly see it's there. So one possibility could be is that maybe the cell line was already exposed accidentally to vaccine, or not vaccine, maybe it would have been the real virus, and perhaps virus might have inserted itself into the genome. So you can see that's not addressed by the authors at all. I would have liked to see what causes these. We're going to now go see other figures right here. And in this figure, what they're showing is that when they took the cell line and exposed that cell line to vaccine to different amounts of time, different amounts of vaccine for different periods of time, this is what the 6 hours, 24 hours, 48 hours mean, they saw different amounts of activation of that selfish gene. That's the line 1 gene that I was telling you about a moment ago, and they see different amounts of activation. And they proclaim that vaccine itself, the genetic material of the vaccine itself can activate this selfish gene, and they make that claim based on this observation that you see with the green dots. So when the cells were exposed to 6 hours, largest amount of vaccine that they introduced to those cells did indeed show more genetic expression, or basically use of that gene more frequently than in controls. My problem with this observation is that as you move to 24 hours, you can see controls have more of that gene being used. And then if you look at it 48 hours, it's all over. The control has the highest amount and it's huge. So the singular observation that they see right here, that vaccines activate this gene, I don't necessarily agree with this, because it just might be random, because as you can see, this gene is activated under any circumstance. And in fact, I would say that vaccines perhaps suppress the activation of this gene, and only in this initial stage, for whatever reason, they might have an increase of the use of this gene. Now, they're suspecting that it is this gene that then what it does, it converts somehow, that's not explained how, but somehow that gene, the product of this gene, might be grabbing the RNA of the vaccine and converting it back to the DNA. And they show it in the last figure, not this one. This one shows you once again the protein, the protein line 1 protein product in the cell. So blue is nucleus, red is the protein molecular robot of that selfish gene. You can see it's all over those cells. And here they claim vaccines seem to be activating it. This is only when they expose it for 6 hours. So that's replication of the previous slide. And this is the final image that they show. What you're seeing here is they did a PCR amplification again, but now directly on the DNA. So they isolated all of the DNA from these cells, and they amplify a specific element of interest. Again, just like the PCR test that is used on all infected patients. And they were able to show that they can copy a specific genetic DNA fragment that corresponds to the exact sequence as the vaccine, which means that vaccine had to be converted back into DNA. And what you see here is then they took those copied fragments and they ran it on a gel, and basically the DNA weaves its way through the gel, and then bigger fragments run slower, smaller fragments go faster. And this is why you can see here is bigger fragments are on top, smaller DNA fragments can go through that gel faster, so they're at the bottom. And they show DNA. That's what these lines are showing. It's the DNA that they copied over and over that corresponds to DNA sequence that is identical to the vaccine, which normally is an RNA. And that's their proof that the vaccine can be converted back into DNA. But my problem with this proof is, this is definitely, we can clearly see they're showing DNA. But my problem is the fact that their initial figure right there, this one, the control, in those controls, they were able to show they can also isolate that same sequence from these cells, even though they were never exposed to the vaccine. So once again, where does it come from? So something is amiss, they don't address it, and perhaps the DNA that we're seeing right here, that they claim, hey, we have this DNA, we see this DNA in these cancer cell lines, perhaps it's because of whatever was producing this signal that should not have been seen in control cell lines, meaning either those cells were already contaminated with the vaccine, or contaminated with the virus, and the virus was present inside there as well. We need to have this addressed before we can make claims with 100% certainty that vaccines can be converted back into DNA. Nevertheless, this is the first such observations that has been demonstrated, very important. So how about the fact that normally the selfish gene is supposed to be grabbing its own RNA and not other foreign RNAs? So how are we getting the DNA from vaccine? Well, there was actually a paper published in 2018 where authors of that publication showed that this selfish gene is also present within platelets. Now, platelets are an unusual type of cells because they don't have nucleus. The way that platelets are formed is they bud off from other cells, and they do not have nucleus because their function of platelets is pretty much heal the wounds and stop the wound from being able to bleed. And these authors showed that platelets also have line 1, and the authors also have shown that the selfish gene inside platelets can grab RNA that resides inside platelets that are not line 1 RNA, meaning they can grab RNA of other that are products of other genes, and they can make hybrids between RNA and DNA. So this selfish gene reverse transcriptase inside platelets appear to have own normal function where it can grab many different RNAs and copy it into DNA, and then you have a hybrid between both DNA and RNA. And these authors proclaim that perhaps this is platelets' own regulatory mechanism of ensuring when the RNA is actually used, because when you have a RNA and DNA hybrid, that RNA can no longer be used as a template to produce your protein of interest. So this is the first time these authors in 2018 proposed completely new way of potentially how our genetic material could be regulated. And it could be regulated through this selfish gene. Perhaps this is another reason why the selfish gene has also been retained throughout evolution. But this is something that we've never known before, and it's been demonstrated only for the first time in 2018. I'm not even sure if it's been confirmed since then, this completely new mechanism of how our genetic material could be regulated. And what's interesting is that they even claim that this could be the reason why HIV patients have an increased risk of having blood clots. And the reason why is because HIV patients, as I already told you, they have to take reverse transcriptase inhibitors in order to survive, in order to make sure they can survive the HIV infection. But as a consequence of that, the authors of that 2018 publication claim when they inhibited that reverse transcriptase, then the platelets were no longer allowed to work normally. And this distorts the proper function of the platelets inside HIV patients. And this is why perhaps these then platelets can contribute to the development of clots. The reason why this is fascinating, because perhaps this could be the mechanism how we are observing increased likelihood of clots inside vaccinated people. Perhaps under some unusual circumstances in some individuals, the vaccine does get inside platelets. Remember, it's supposed to stay inside your muscle, but some of it might be getting inside the platelets. And when the vaccine gets inside the platelets, well, according to this publication we're discussing today, that vaccine mRNA can interact with this line 1 selfish gene product, and perhaps that prevents that product from properly functioning inside the platelets. And this is once again how the proper platelet function is distorted, and why some vaccinated individuals might have increased risks of having clots. So that's a very unusual link. I don't think anyone has mentioned this link yet. So here you go. We're just gonna propose it for the first time here. And perhaps that could explain why, if it is indeed possible that the selfish gene product can indeed interact with the mRNA vaccine. Now, what does it mean? Could it mean that you could use the vaccine mRNA and introduce it back into the genome? Well, in theory, yes, because that's exactly how the selfish gene reintroduces itself back into the genome. But that has not been confirmed at all by this paper. So we do not know if that can be true. And then another important thing is, even if it were to be true, then remember, this was inside cancer cells, not normal cells. Activation of the selfish gene is not normal inside normal healthy cells, with potentially exception of the platelets. All right, so even if it were to be true, then it would have to be in unusual circumstances where the function of this selfish gene product line 1 is dysregulated, and that appears to be more under situations if there's a cancer happening or other potential diseases, because sometimes this selfish gene is activated in certain disease developments. So something that we need to study further, investigate further. Definitely now very important to be testing this and looking at this in more detail in case this could turn out to be true. Right now, we do not know though. All right, so that's the final conclusion. All right, now let me tell you quickly about the event that we have upcoming, COVID-19 Q&A number 6. The way it works, we collage a number of questions from YouTube or emails, and we answer top 10. And then it's open mic to everyone. We grab questions from the audience, or we have a discussion. It's supposed to be a safe environment. The first 10 people who will subscribe to my newsletter will give you free tickets away, so you can come and join our next event. And the link to the newsletter subscription is in the description below, so check it out. Another thing is, we have an ongoing survey going on where you, the audience, can decide the future topic videos, and we propose 6 new topics. So please check out the survey. The link to the survey is also in the description below. So far, it looks like T-cell exhaustion is the main topic of interest, but go ahead, please vote. Let us know what you want to learn in the future installments of these videos. We love the fact that audience can tell us and direct us as to what kind of information you would like to hear in the future. All right, so then if you like this video, please give us a like, subscribe to the channel, share the video so that we can grow. And we are looking forward to seeing you next time. Bye, everyone.